So today, Professor Stuart Hameroff is with us, and he is the father figure of microtubule research for information processing. In 1982, he proposed for the first time that microtubule processes information, and after 1994-1995, he moved on to the claim that microtubule is responsible for consciousness after collaborating with Professor Roger Penrose. And so two big journeys he have traversed in the last 40 years with microtubule, and today he would enlighten us on consciousness and happiness and different aspects of consciousness and his journey through different works of on the microtubule and consciousness and other aspects. And he organizes one of the biggest conference on consciousness in the world since 1994-1995, and it is still ongoing. And then he is with us today, and he is making this presentation for the exclusively for the SPARC project. He is the principal investigator, and the project is management of fractal time in the management of complex decision making. So to Professor Stuart Hameroff.
Thank you, Anirban. Thank you very much. It's a great pleasure to be here. Hello, everybody. I'm going to talk about consciousness, and particularly in relation to happiness. And if we look at the slide, we take it for granted that when we open our eyes, the world out there appears inside our head. Bing. And I'll use Bing to mean consciousness, experience, qualia, phenomenal awareness, subjectivity. And this is approached through neuroscientists who probe the brain, artificial intelligence people who build robots and try to simulate consciousness, artists who capture the essence of consciousness, physicists who study reality, for example, Schrodinger's cat, psychiatrists who look inside the subconscious, anesthesiologists such as myself who try to figure out how anesthesia works, meditators, and philosophers. Now, most of neuroscience, modern science, thinks of the brain as a computer of individual neurons shown here connected by synapses. And this is likened to a computer network of nodes and variable strength connections. This is based on the Hodgkin-Huxley integrate and fire neuron, which goes back to the 1950s. We know that neurons consist of dendrites and cell body, which receive inputs at synapses from other neurons and transmit information signals along the membrane through ion channels, and integrate this information at a threshold, which when reached triggers axonal firing or spike to the next neuron, the next layer. And this propagating signals are all along the membrane. So integrate in the cell body and dendrites, fire along the axon. Integrate, fire, and this can be put together to make a computer or a model of a computer. Now, the prediction of Hodgkin-Huxley was that there's integration in terms of voltage in the dendrites and cell body, which when reaching a threshold, a fairly narrow threshold, triggers spikes shown in red, which then travel down the axon. And here we see it schematically with a very narrow threshold in voltage and a very narrow threshold in time, and then signals moving along the axon by a sequence of ion channel opening, which is why the slope, because it's moving in one direction. This is from the work of Nondorf, Wolf, and Volgashev in Nature in 2006. This type of processing would be deterministic, algorithmic, no place for consciousness or free will. However, what's actually seen, and Nondorf et al. Implanted electrodes into pyramidal neurons of awake animals, of awake cats, and what they found was a very wide threshold that could vary from spike to spike and over time. Wide threshold, wide time. So there's something else mediating the triggering of axonal spikes, which exert behavior. And this is a very good place for consciousness to come in and regulate spikes other than what's happening at the membrane potential. So this is where Bing could happen, consciousness in terms of this variability, influencing spiking, and over time here. So where consciousness could regulate behavior in the adjusting the threshold of spiking in dendrites and soma. Where could this come from? Well, I think it comes from a deeper level inside the neuron. So if we look at a neuron, here's the synapse and here's the membrane of the dendrite and cell body, and then the axon. And inside we see microtubules shown here, and there's actin and microtubules, and the microtubules and dendrites are mixed polarity and interrupted. They're short and broken in a mixed polarity, one pointing one way, one pointing the other way in networks. That's unique in biology, in dendrites and soma of neurons. And once you get to the axon, they're continuous and go all the way to the synapse. And the question is, does consciousness come from membrane effects on the surface, which is what classical Hodgkin-Huxley would explain, or from the microtubules inside? And for various reasons, I think it's from the microtubules inside, as we'll come to in a second. Now, if you look at the microtubules inside a neuron, they do many things. For example, here's a dendrite, here's a neuron with a cell body. Here's the axon going this way. This is one particular dendrite. So materials in synaptic plasticity, in adjusting synapses, say here, or somewhere downstream, are synthesized in the cell body and then transported along microtubules by these motor proteins, kinesin and dynein, which move along carrying cargo to synapses to adjust the synaptic strengths. And at some point, when they come to a synapse that needs material, they jump off and deliver their cargo. So the question is, how do they know where to get off? And they have to go from microtubule to microtubule, and then at branch points to decide whether to go right or left. It turns out that this protein tau actually signals the motor proteins to get off and deliver their cargo. So the placement of the tau protein on the microtubule is a code, like for memory, because it tells the motor protein where to deliver its cargo to adjust synapses. So the placement of the tau on the microtubule is a form of memory. Now, if the microtubule falls apart, disassembles, or the tau falls off, then you get Alzheimer's disease. Now, Alzheimer's disease has amyloid plaques outside the neuron, but the key to the loss of memory and cognition appears to be the neurofibrillary tangles, which are the tau proteins, which are associated with the disassembly and destruction of the microtubules. And this seems to cause the loss of cognition, and then the whole you lose the synapse, and the whole neuron kind of shrivels up, and you lose neuronal mass. One thing about memory is modeled by long-term potentiation, which means that the synapse is strengthened with repeated use. And one thing, and how that happens is unclear. Now, most people would say it's because the membrane proteins are adjusted, but the membrane proteins last hours to days, and yet memories can last lifetimes. So it needs to be encoded. Memory needs to be encoded at a deeper level. And one thing that happens is that calcium is released and activates an enzyme called CAMK2, this hexagonal protein shown here, which then attaches to microtubules. Now, and can phosphorylate microtubules or other materials inside the cell. CAMK2 is a snowflake-shaped hexagon, which with calcium gets activated, so these kinase domains spring up and below the main body, and they have phosphorylation sites shown here. So there's 6 below and 6 above that can phosphorylate something that could be a form of memory. And we noticed, along with my colleagues Travis Craddock and Jack Tusinski, that the size and geometry of the CAMK2 exactly fits the size and geometry of the microtubule A lattice and B lattice, and we suggested this as a mechanism for memory in this paper in 2012. So what we think is happening is the CAMK2 lands on the microtubule lattice, and each of the 6 kinase domains can deposit, can phosphorylate, and essentially depositing a bit of information. So up to 6 bits of information can be encoded by each CAMK2, and this could be for tau binding or other forms of memory. This brings up the larger question of whether microtubules may be processing information and acting as computers. And As Anirban mentioned, I got interested in this actually in the 70s and published in the 80s. And here's the structure of a microtubule. It's a hollow cylinder of about 13 protofilaments of tubulin, a peanut-shaped protein. And originally we thought the protein might change its shape to represent information. It turns out that's not necessary, but I'll come back to that point. But we model this as a cellular automata, such as the game of life. Here's the game of life on a square grid, and just with simple neighbor rules, you can get patterns moving through the microtubule lattice, or through the grid in the game of life. We applied it to microtubules, which are a hexagonal skewed lattice, and we found similar patterns able to propagate through the microtubule lattice and store and process information. So this is the basis for information processing in microtubules initially. There are some problems with this, but which I'll come back to. But basically, I published a bunch of papers in the 80s, 90s, and subsequently with Rich Watt, and I wrote a book in 87 about it. Steen Rasmussen at Los Alamos, where we put two microtubules in opposite polarity together and showed they could show learning in microtubule networks. Jack Tusinski, Travis Craddock, and others. And then in the mid-90s, I started work with Roger Penrose on quantum models of computing, which in microtubules, which I'll come back to. Now, one thing is that this implies a much greater computational capacity for the whole brain when you go down to the level of microtubules. So, for example, the standard idea in artificial intelligence and singularity is you have about 100 billion neurons with 1,000 synapses each neuron operating at about 100 hertz. Gives you 10 to the 16th operations per second per brain. And Kurzweil and others said, well, when we reach this, we'll have brain equivalents in a computer and therefore consciousness, because the brain is obviously a computer. Well, it's not obviously a computer, and this has actually been reached, I think, and there's no conscious computers as yet. If we go to the microtubule level, where one tubulin equals 1 bit, and these tubulins are oscillating at about a 10 megahertz, then you get 10 to the 16th operations per second per neuron at the level of the microtubules, and 10 to the 27th operations per second per brain. We do it a little bit differently with quantum dipole pathways, and I'll come back to this. You lose a little computational capacity, but you gain in terms of stability against decoherence. But it's still much greater than it would be for one bit per neuron. But still, where's consciousness? Even at this incredibly more vast computational capacity, where's the Bing? Where's consciousness, even at microtubule computation? Is something else required, such as quantum physics? So this is what I faced in the early 90s when somebody asked me, well, let's say you're right, how would that explain consciousness? And I didn't have an answer. I was faced with the hard problem, as it later became known. But fortunately, this person suggested I read Roger Penrose's book, The Emperor's New Mind, which I did. Computers, Minds, and the Laws of Physics. And in that book, Roger used Gödel's theorem to show that conscious understanding is non-computational, that something outside the computational system is required for knowing or understanding, and that something is most likely to be involved with quantum state reduction, with collapse of the wave function, and the measurement problem in quantum mechanics. And further, that this was a factor related to the underlying structure of the universe, space-time geometry, and quantum gravity. This was very confusing to me when I read it, and the more I pondered it, it began to make sense. And this introduced a non-computable influence and gets us out of the trap of being completely deterministic and algorithmic. So this is a lot to understand here. So let me try to unpack it for you. Roger began by addressing the fundamental question of how particles can be in quantum superposition of two or more locations simultaneously. In a quantum computer, information can be not only as 1 or 0, but can be in quantum superposition of 1 and 0. This is one of the strange things about quantum mechanics, that things can be in two places at the same time. But until you measure or look at them, and then they seem to collapse or reduce to one or the other. We only see or observe definite states. We don't see the superpositions. And that's called the measurement problem in quantum mechanics. But before we even get to that, how can something be in two places at once? It's still kind of very, very counterintuitive. Roger explained it by combining quantum physics with general relativity. Now, Einstein's general relativity had shown that matter was equivalent to curvature in fundamental space-time geometry for large masses like planets and the sun, that there was this background space-time that was curved due to the gravity, or was equivalent to the gravity of these giant masses. And so, for example, he predicted that if we're here on Earth, that a star that would be behind the sun could be observed because the curvature of space-time by the sun would bend the light, and that we would see this star over here even though it was actually behind the sun. This was a testable prediction. And in 1919, Sir Arthur Eddington tested this and proved it by going to the top of a mountaintop during an eclipse, and he could see the stars that were behind the sun, actual position, which were apparently over here, and they became visible, and all the other stars were shifted by the amount that was predicted by Einstein's general relativity. So this confirmed Einstein's theory, and here are Einstein and Eddington congratulating each other since they both won Nobel Prizes. Einstein for something else, but Eddington for this. So that was a great achievement, but what about matter and space-time curvature at very tiny scales? Only Roger Penrose, as far as I know, has addressed this issue, and he began by illustrating space-time, four-dimensional space-time, 3 dimensions of space and 1 dimension of time, as a 2-dimensional space-time sheet, where space was condensed to one axis and time on the other. So a particle here would be a curvature over there, and a particle moving over there would be over here, so that an oscillation of a particle between two positions would be an oscillation of space-time curvature. And one could imagine that superposition of a particle being in both places at the same time would be both these curvatures. So the particles here and here, because we have two curvatures. So there's actually a separation in space-time geometry that can give rise to curvature and two curvatures for the particle. Now, excuse me, the so-called Copenhagen interpretation, in which conscious observation causes collapse, would be something like this. This is Schrodinger's cat, where you have a superposition of both states until somebody opens the box and then looks. And this was a popular explanation, which conscious observation, Bing over here, causes this one to cease to exist and selects this one to continue. So this would be the dead cat or the live cat, or any type of superposition. And but Roger Penrose had a different explanation, which I'll come where a threshold would be reached called at time t equals h bar over e sub g, at which moment collapse would occur along with a moment of consciousness, and I'll come back to that. You could imagine that if both superpositions continued, each would form its own universe, and we'd have multiple worlds. And this is the basis for the multiple worlds hypothesis, in which the universe, there is no collapse, and each possibility continues to evolve. But Roger said that superpositions, separations, would be unstable and self-collapse, that is, undergo objective reduction at time t equals h over e sub g. So at this time here, t equals h bar over e sub g, there's a moment, this one ceases, this one continues, along with a moment of conscious experience. So rather than consciousness, as in the Copenhagen interpretation, causing collapse, Roger said collapse occurs spontaneously and causes consciousness at a time t equals h bar over g. And so rather than consciousness causing collapse, collapse causes consciousness. Now these moments occurring in the environment all around us, which might look like decoherence, would be proto-conscious, because they would be isolated, random, they would lack meaning, they would be occurring in the atmosphere and the environment all over the place all the time. Moments of proto-consciousness. So there would be some experience there, because experience is an intrinsic feature of the universe at this level of space-time geometry. But they wouldn't have any memory, they wouldn't have any meaning, they wouldn't be connected to anything. So they wouldn't have any impact, and they would be occurring all over the place. Kind of like panpsychism suggested that everything is conscious, but panpsychism is a state of a particle, a property or state of matter, of particles, And this is before that. This is the collapse actually selects those particular states. So it's more fundamental. So they would be proto-conscious, similar to the philosopher Alfred North Whitehead's simple occasions of experience. Now, as a metaphor, you might think of these proto-conscious moments occurring everywhere, but not having any meaning, like the notes, tones, and sound of an orchestra warming up. If you go to a symphony, every musician is tuning his or her instrument, and you hear all these different tones and notes, noise really, a cacophony. And what is needed for example, in the brain to make consciousness as we know it, something that would orchestrate these proto-conscious moments and create something analogous to music, something that would be organized, rhythmic, make sense, have continuity, have meaning, and so forth. And that's what Roger was looking for. How could these proto-conscious moments, due to these very tiny ubiquitous collapses, be organized, orchestrated in the brain for full, rich, conscious experience? So Roger needed a quantum computer in the brain which could biologically orchestrate quantum information processing, halt or terminate by Penrose objective reduction, connecting to space-time geometry and non-computable platonic values and qualia, that is awareness and feelings, and regulate functional neuronal and synaptic activities so that consciousness could actually exert behavior and have an effect on behavior. So I thought when I read Roger's book that microtubules were what he was looking for. And he didn't know about microtubules. He knew that neurons were too big to be quantum objects. But if you look inside and see the microtubules, they have much more promise. So we met in the early 90s, and this is at the Grand Canyon after the first the Science of Consciousness conference in Tucson. There's Roger, there's me, and there's Dave Chalmers, the philosopher, and we began to discuss this and put a model together. So the questions, the problems we faced were the following. To answer whether neuronal brain microtubules could orchestrate objective reduction to provide conscious moment which regulate neuronal firing and synaptic plasticity, and this led to the ORch-OR theory, orchestrated objective reduction. So OR is Roger's self-collapse of the wave function, and we were suggesting that microtubules in the brain could orchestrate these OR events to give rise to full, rich consciousness, like an orchestra converts noise to a symphony, or to music. So we first had to define the quantum bit or the qubit in microtubules. Again, in a quantum computer, you have a qubit is where you have a superposition of states. So instead of 1 or 0, you have 1 and 0, and then they collapse to either one or the other. That would be a quantum bit or qubit. So we had to define that. We had to show how to apply E sub g equals h over t to microtubules to quantify ORch-OR conscious moments. And we wanted to test sensitivity of these processes to anesthesia, which erase consciousness selectively, and also psychedelics, which might enhance consciousness, and show how ORch-OR could account for EEG and other correlates of consciousness. So Roger had essentially described a space-time qubit, a quantum bit in space-time, and we wanted to connect that to microtubules, initially as a tubulin qubit, but then we moved on to once the structure of tubulin was known, to a pathway qubit, which loses capacity but increases tremendously in terms of stability of coherence. And the idea was that the pi resonance rings inside tubulin would be aligned and couple and oscillate back and forth. And that would be the oscillation. And then when in superposition over here, when that reached threshold, we have a conscious moment. So basically what we want to do is connect space-time qubits with these ORch-OR qubits. And then we'd have sufficient superposition, shown in the gray, in many, many microtubules. We only show one here, throughout the brain, that would reach threshold at t equals h over e sub g, and have a moment of consciousness which correlate with a collapse in space-time geometry at a much deeper level. So in one of our early papers, 96, we calculated the E sub g, the gravitational self-energy required to separate matter or its equivalent space-time curvature from itself in each tubulin by 3 different methods, depending on how the tubulin would be separated from itself. Was it the whole protein that was separated from itself, or was it the level of atomic nuclei within the proteins, or by the nucleons, the protons or neutrons? And we calculated for these three methods, and calculated that the atomic nuclei would have the dominant effect because it would kick in first because it has you require the fewest mass to reach threshold. And for example, for ORch-OR conscious events at 40 hertz, if you imagine that we have an ORch-OR event every 25 milliseconds, t equals 25 milliseconds, you would need 2 times 10 to the 10th tubulins required. And we published that in this article in 1996. There are about 10 to the 9th tubulins per neuron, maybe less, but depending on the fraction in superposition, this would require only a small number of neurons, only about 20 neurons in the whole brain. And the 25 milliseconds was a very long time to avoid decoherence or premature OR. So we then, in 2014, we revised our theory and considered shorter intervals T with much larger E sub G, in other words, faster frequencies, which could give rise to slower interference beats. So, for example, for ORch-OR at t equals 10 to the minus 7 seconds, 10 megahertz, we would need 2.5 times 10 to the 15th tubulins, and this is about 10 to the minus 5 of total brain capacity. It's about 10 to the 20th tubulins in a brain, and quantum coherence for this long in microtubules had been shown experimentally in terahertz, gigahertz, megahertz, and kilohertz frequencies by Anirban and his group in NIMS and SCUBA in several papers in 2013 and 2014. So we had some evidence for this, and here's Anirban's group results showing at 3 different scales, at the level of neurons, the level of individual microtubules, and at individual tubulins, that looking at conductance frequencies, applying electrodes and seeing where the conductance jumped significantly, which would be a ballistic conductance and possibly quantum conductance deeper inside without the interface. And he saw these triplet patterns, triplets of triplets, or octaves as they call them, at terahertz, gigahertz, and megahertz at the level of tubulins, and at the level of microtubules, the same sort of thing at gigahertz, megahertz, and kilohertz, and then at whole neurons, megahertz, kilohertz, and hertz. So we were seeing these self-similar patterns repeating in microtubules and tubulin over many, many orders of magnitude, many scales. So this led to a model, something like this, where we start with a neuron, a pyramidal cell body, and most views of the hierarchical organization of the brain would go higher into neuronal networks and then regions of the brain, but we focus on going lower into microtubules, tubulin pathways, the pi resonance dipoles, and then eventually all the way down to space-time geometry. So space-time geometry would go roughly 10 to the 15th to 10 to the 43rd hertz, and then we get into these pi resonance oscillations in terahertz at 10 to the 12th, gigahertz in the tubulin, 10 to the 7th in microtubules, and so forth, until we got to the EEG in hertz. And Roger suggested that at, say, 10 megahertz oscillations, we might get, if microtubules had slightly different frequencies, we would get beat frequencies, and much slower, that would give rise to EEG down in the hertz range. So this is like when two musical instruments are not exactly tuned, you get beats and feedback, and down in the in this case in the hertz range. So we proposed that EEG, say under 100 hertz oscillations in neurons, was actually coming from microtubules who might be oscillating at a much higher frequency but having interference beats. So we proposed that microtubule oscillations are the origin of EEG. And EEG has been used for almost a century, but we don't really understand its origin and where it comes from and why it's coherent over the brain. So the qubit, as we eventually defined it, was these pathways oscillating back and forth. And this gives stability to the quantum bit because if you have a pathway, it's kind of error correcting. And if one falls out, the other ones will pull it back into to coherence. Now, we looked at, this is actually kind of a euphemism or a simplification of what's really going on. And in this work by Travis Craddock et al. In 2012, we look more closely at inside the tubulin. So here's one tubulin, and it's got 86 of this pi resonance, these pi resonance rings in aromatic amino acids, tryptophan, phenylalanine, and tyrosine. And also in the red, we see where anesthetics bind to erase consciousness, or where anesthetics bind, and we think by erasing consciousness by blocking these dipoles. So we should now talk about these aromatic rings and pi resonance, because they are the basis for organic chemistry, and very important in life and in consciousness. So in about the 17th century, chemists knew that the hydrocarbons, chains of carbon with hydrogen, had a formula, basically CnH2n plus 2, which would be the alkanes, linear chains of single bond carbons with hydrogen, or alkenes, where you had one double bond, CnH2n. But they also knew about C6H6, but they didn't know its structure. And they pondered it, and one night Kekulé, a Dutch chemist, had a dream that these were like snakes. These chains were like snakes, and the one snake swallowed its tail, and this is known as the Ouroboros in mythology, and that this might be benzene, and benzene could be perhaps a ring. And this was the first time anybody had thought of that. And sure enough, he was right. And benzene is a ring of 6 carbons, and with therefore 3 extra electrons. Now what that means is that the extra electrons are these pi resonance that would stick up above and below the ring, but they coalesce and eventually form an electron cloud. And these 3 delocalized pi orbital electrons among 6 carbons form electron clouds conducive to quantum effects. In these clouds, you can have electric and magnetic dipole oscillations, excitons, plasmons, charge transfer, phonons, fluorescence. So this would be a quantum-friendly region. If we have 2 of these benzene or phenyl rings, they attract by van der Waals attractions and induce dipoles. So the electrons here repel the electrons there, so we have these clouds with a dipole, electron cloud dipoles, which then oscillate back and forth in terahertz. And they can also form superposition of being in both of dipoles pointing in both directions. This is very important in biology. For example, protein folding. If we have these amphipathic molecules with a benzene ring and a polar tail, the non-polar benzene rings will attract each other to form micelles, precursors to proteins, and or proteins themselves actually, which have these non-polar pi resonance interiors and polar water-soluble exteriors. And because that's what amphipathic molecules have, a non-polar interior and a polar exterior, and they form these micelles, and this is how proteins fold. Also, lipid bilayers have the non-polar groups in the middle and polar on the outside, and nucleic acids, DNA, RNA, have the pi resonance groups there. So proteins, lipids, nucleic acids all have an interior region of non-polar pi resonance groups that can support quantum effects, and so we call this the quantum underground inside underlying biomolecules, pretty much all biomolecules. It should also be noted that molecules that have psychoactive effects, the neurotransmitters dopamine and serotonin, and the psychedelics LSD, DMT, and psilocybin, have these pi resonance clouds, very large one in the case of LSD. So they seem to have something to do with consciousness. Now the opposite would be drugs that inhibit consciousness. For example, anesthesia, anesthetic gases, for example. And during the 19th century, a group of gases were discovered which, when inhaled at low concentrations, caused euphoria. And this was known as ether frolics. People sniffing just a little bit of ether got all happy and giddy and danced and acted crazy. And similarly with laughing gas, they would get all happy and giddy. But if you keep breathing more, at higher concentrations, you are rendered, all humans and all animals were rendered unconscious, that is, unresponsive. And this led to anesthesia and the first use of anesthesia for a surgical procedure rendering a patient unconscious at the Mass General Hospital in 1846. And so from there, anesthesia became a useful tool for surgery. Now, anesthesia is very interesting. The studies showed that the anesthetic gases spared non-conscious brain activities, affecting only consciousness. So they're fairly selective for consciousness. And anesthetic potency, the minimal alveolar concentration, or MAC, would be the average concentration needed for any particular gas to render subjects unresponsive, varied among the gases, but for each gas was the same for all animals: insects, humans, salamanders, horses, and also plants respond to in terms of their cognition of tracking the sun, for example. That can be anesthetized. And anesthetic gases varied in chemical makeup. They could be ether, halogenated hydrocarbon, or the inert gas xenon. So here are 10 anesthetic gases, and they're all different structures. Here's xenon, there's some ethers, halogenated hydrocarbons. What they have in common is, shown in the gray, nonpolar regions, kind of like pi resonance, but of nonpolar regions, which seem to exert the anesthetic action by van der Waals London dipole dispersion forces. This is a quantum interaction by these uncharged nonpolar groups, which can bind to each other, much like the pi resonance couple and oscillate. And they would do so in a specific solubility medium in the brain, because oil and water don't mix, and they're oil-like. So at the turn of the 20th century, scientists sought a common factor which correlated with potency, that is 1 over MAC. The more potent, the less you needed, so it would be 1 over MAC, and search for the proper solubility phase in which anesthetics dissolved or bound in the brain and body. And two scientists, Hans Meyer and Charles Overton, working separately, studied a whole bunch of gases in a whole bunch of animal systems to determine their potency, and then looked for a solvent in which might correlate with their potency. And they found one in olive oil. And what they found was that the more potent a gas was in olive oil, for example, methoxyfluorine, the more potent it was in putting animals to sleep. So, for example, methoxyfluorine, you needed a very tiny amount, 0.25%. Now, something like nitrogen, which we breathe all the time, would be an anesthetic, but at about 50 or so atmospheres. So only at very, very high, much higher than 1 atmosphere. And they fell along this line over many, many orders of magnitude. A very curious thing, because they acted on all animals and were the same for each animal, although the gases were different. So depending on how potent depending on how soluble they were in olive oil, they would be more potent. Now, what does this mean? If you think about taking a body or a brain and looking at its what materials it would dissolve, its solubility phase, if you grind up a body and look at the different solubility compartments, you get something like this. So polarity goes this way. So out here would be water and blood with a lot of polar charges, and as we go this way, we get nonpolar. And what was determined was that anesthetics bound in these highly nonpolar aromatic regions, which turned out to be chemically dimethylbenzene, methylbenzene, and benzene, which are the pi resonance groups that I mentioned earlier. So this is where anesthetics bind, and therefore where consciousness should arise from. So, for example, we have these dipole oscillations oscillating back and forth. The anesthetic comes in and forms its dipole dispersion and it dampens the oscillation, and that causes unconsciousness. So this is required for consciousness. You need the superposition also. And then the anesthetic comes along and disperses the dipole and dampens the oscillation. So anesthetic gases bind non-specifically and act by these weak quantum forces and bind in nonpolar regions, for example, fat sites all over the body. It's a very, very non-specific type binding. And Christoph Koch, a famous neuroscientist many years ago, said that anesthetics could not be useful to determine consciousness because they're too nonspecific to shed light on consciousness. And in fact, they do bind anesthetics. They do bind nonspecifically all over the place, but they block consciousness selectively and affect very little else. And the reason is, I think, that the systems sensitive to anesthesia must be highly organized quantum systems, thus easily perturbed. So only a system that has highly coordinated, highly orchestrated quantum processes would be affected by anesthesia, where if you perturb just one part of it, you mess up the whole thing, like a Bose-Einstein condensate. So I think this explains why anesthesia is selective, despite binding very non-specifically. So where in the brain might this happen? At first, it was thought that anesthetics bind in lipid regions of membranes, but then in the 1980s, Franks and Lieb showed anesthetics act directly in proteins in nonpolar hydrophobic pockets, for example, in the firefly luciferase. And here you see the pi resonance group, and this is where the anesthetics would get in, and these were called hydrophobic pockets composed of pi resonance groups, including the aromatic amino acids I mentioned earlier. So people thought, well, it must be membrane proteins, ion channels, receptors, and so forth. So membrane protein ligand-gated ion channels became the presumed sites of anesthetic action. Anesthetic gases bind to these membrane proteins, for example, receptors for serotonin, glycine, acetylcholine, and GABA. But experiments on membrane proteins produced a mixed bag of conflicting results. Some anesthetics open a channel, others close it. Not all anesthetics bind to any one receptor. So that doesn't appear to be the answer. Around 2008, Eckenhoff's group, University of Pennsylvania, did a systematic search and found that 70 proteins bound anesthetic in a neuron, about half in the membrane and half in the cytoplasm. And they then narrowed that down and looked for genetic expression for genomics and proteomics, and found 3 proteins common to both, tubulin and microtubules, heat shock proteins, and another enzyme, and only tubulin is in the signaling pathway. So the proteomics and genomics pointed to microtubules, according to their work. And they also did a study using an anesthetic fluorescent anthracene, which is only an anesthetic when you illuminate it with ultraviolet. And they gave it to tadpoles who have conveniently transparent heads. And so they gave it, and when the tadpoles were swimming around, they illuminated with ultraviolet light, and at that moment the tadpoles stopped swimming and went belly up. They went upside down basically. They were anesthetized, and they noted this was consistent with our theory, which they call quantum mobility theory. And then when they ground up the tadpoles, they found that the anthracene was bound to microtubules. So this pointed to microtubule action of anesthetic gases. So to kind of what we then did, what Travis Craddock and our other co-authors did, was zoom in on tubulin a little bit more. And as I showed you earlier, each tubulin has 86 of these pi resonance rings, and we know this is where anesthetics bind. So we modeled quantum dipole oscillations among all 86 pi resonance rings in tubulin, and we found a spectrum of oscillations at KT, ambient temperature, a spectrum of collective dipole oscillations with a common mode peak at 613 terahertz in the blue-green light region of the spectrum. Right about there was the common mode peak, and this was the spectrum we saw, and included the anesthetics that we sampled also. And we then added the anesthetics, and we found the presence of each anesthetic gas abolished the 613 terahertz peak and dampened other terahertz frequencies proportional to anesthetic potency. So the 613 terahertz peak would be here going up, and in the case of desflurane anesthetic, it turned negative, so it wasn't there. Ether turned negative, same with, or it was absent. I'm sorry, these are subtraction spectra, so they show you what is missing. So what's missing from the normal spectrum in case of desflurane includes the 613 peak. It's missing with ether, it's missing with enflurane, it's missing with halothane, isoflurane, methoxyflurane, nitrous oxide, sevoflurane, but it's not missing, and also fluorothil, which is an anesthetic anticonvulsant, but it's not missing in these two molecules, F6 and TFMB. And these are non-anesthetics. Now, the Meyer-Overton correlation has a few outliers, including TFMB and F6, which bind in olive oil. So they're binding, here's TFMB, would be here, it'd be a pretty good anesthetic. In fact, a very potent anesthetic. F6 would also be a good anesthetic, but they don't. They have no anesthetic potency. So I put the MAC as 1,000 or infinite. You need an incredible amount to have any effect. So why are they not anesthetic? They bind to the site, but they don't cause loss of consciousness. They had no effect on tubulin terahertz. So they didn't affect it at all, and I showed you in the previous slide. So why not? We then tested their polarizability, and we found that TFMB and F6 were more polarizable than any of the other anesthetics. So what we think that means is that an anesthetic, so here's the dipole oscillation, and an anesthetic would form dipole dispersion forces and put drag on the oscillation and stop the oscillation. But the non-anesthetics are more polarizable and kind of go along for the ride. They bind and oscillate right along with the protein, and therefore don't exert any drag on the oscillation and don't cause anesthesia. So they are highly polarizable and may go along for the ride, so to speak. Now, in conclusion on this part, the anesthetic gases abolished and non-anesthetics did not abolish the 613 terahertz blue-green peak and intraneuronal correlate of consciousness here. So it's interesting that blue-green light may be the origin of consciousness in the brain. So here again is the model that we have of going down and where the oscillations in tubulin give rise to consciousness by reaching E sub G equals H over T, and that the anesthetic disperses the dipoles and prevents that from happening. So moving back up as a more macroscopic level, where in the brain might this occur? We know that conscious perception occurs several hundred milliseconds after sensory input. So if there's input from the eyes or the ears or the sense organs, it goes to the thalamus, and in the case of vision, to the back of the brain, to the front of the brain, and then spreads out from there. And it's this third wave that seems to be conscious, bing, and only the third wave is affected by anesthesia. So perhaps only the third wave is using quantum mechanisms. Once in the cortex, the signals go to the layer 4, then they go to 1, 2, 3, and 6, and then they converge on these giant pyramidal cells in layer 5. And I've showed you pictures of those before. So it seems that the giant pyramidal neurons might be the best bet for where consciousness can occur in the brain. Not only in pyramidal cells, but if you had to pick one, that's the one I'd pick. Now, interestingly, in these giant pyramidal cells, they have mixed polarity networks of interrupted microtubules. So the microtubules in dendrites and soma are not continuous. They're broken and interrupted. Now, they're part of the cytoskeleton, so it seems kind of strange. If you want structural support like a skeleton, you would have them broken and interrupted. You wouldn't break your bones to get structural support. And also they're mixed polarity, so they're pointing up and pointing down. And this might mean that because they're in different polarities, and they're in a common voltage, for example, from the membrane, that they would have slightly different oscillation frequencies for interference beats, or the origin of EEG. And that could give rise to, as I said, interference beats, and explain where you get the EEG in that interference negative resonance going up to much slower oscillations in the EEG range. And also, EEG does come from apical dendrites, which arise vertically to the cortical surface. So part of the ORch-OR theory is that EEG comes from interference beats of microtubules. So let me discuss something a little bit different and ask, when did consciousness arise? Here's a sort of history of the universe from the Big Bang up until 2000 at least. And when did consciousness arise? Did Bing happen with tools and language, as some people think, fairly recently? Did it happen with animal cells? Did it happen with life, any life? Or did it happen actually before the Big Bang, as some spiritual people would suggest, or in Rogers' cyclical conformal cosmology, in which the Big Bang was preceded by another eon and another, the consciousness might actually come from previous eons and continue. But let's just focus in on one question. Which came first, consciousness or life? And despite a lot of functional descriptions, we don't really understand life. But which came first, consciousness or life? Neuroscience, psychology, and most Western philosophy would say that life was there first. Life preceded consciousness. Consciousness emerged from life. Pan-protopsychism, naturalism, Eastern philosophy, spirituality, Whitehead, and our theory, ORch-OR, would suggest that consciousness preceded life, that consciousness was there first, and that prompted life, and that life actually emerged from consciousness. So how could that have happened? Well, we believe that life on Earth began in a primordial soup proposed in the 20s by Operin and Haldane, a simmering mix from which biomolecules emerged, or perhaps thermal vents. But it's a mixture of oil and water, oil and many other things. And in the 50s, Miller and Urey simulated a primordial soup and found organic amphipathic molecules. They reproduced the conditions thought to exist in the primordial soup, including an atmosphere, lightning, sparks, and materials that were known to exist. And when they looked at what they found these amphipathic molecules, benzene rings with a polar tail, much like dopamine, amphipathic molecules with nonpolar rings and water-soluble tails. And as I showed you before, they don't mix and they coalesce to form these micelles, as Operin described. And Operin suggested micelles were the origin of life. This is also the way that proteins fold. Now, some proteins have a lot of nonpolar pi resonance rings, and you can imagine a coalescence of many of them, perhaps in flat sheets like in graphene. But it could be, and I've suggested this in a chapter I wrote, that in the primordial soup, the nonpolar groups reach threshold by E equals H over T, or T equals H over E sub G, and reach the threshold for a conscious moment, a proto-conscious moment at this time T, and Bing occurred both in space-time but also in this molecule. Now, some of these would have some qualia or feelings of primitive OR events, and they would be random, but some would be positive. That is to say, you could have feelings that would feel good back in the primordial soup. And if that were the case, the feelings of feeling good would serve as a feedback fitness function for systems to develop that would attempt to optimize feeling good. The whole idea of evolution is that self-organization led to promote gene survival, but there were no genes back there. It was probably billions of years before there were genes. So something had to drive organization of life, and I think it might have been seeking pleasure. Now, we do know that these pi resonance rings have two stable arrangements, a T shape and a parallel displaced. So if they're in a superposition of both, perhaps they're going to collapse to one or the other, one of which would be not feeling good and the other one would be feeling good, and the system would use this as a feedback fitness function to optimize feeling good. So in tubulin we have 86 of these, so you have 86 factorial interactions, so there would be a lot of possible feelings even in one particular protein. This is obviously somewhat of a joke by using emojis, but feelings could come from the arrangements of these pi resonance rings, that when they collapse to these particular configurations, you get particular feelings. So with pleasure as a feedback fitness function, orienting pi resonance groups, did life then evolve to orchestrate and optimize OR-mediated pleasure? I call this the quantum pleasure principle after Freud's Beyond the Pleasure principle. And it's true that Darwinian evolution is a pillar of modern science, but the notion that life evolved behavior to promote gene survival is merely an assumption and doesn't really make sense. Behavior is driven by reward. Laboratory experiments, our own behavior is all about optimizing pleasure. Now, it could be a delayed gratification, it could be altruistic, it could be spiritual, it could be hedonistic, or it could be avoiding pain. But we're driven by feelings. Animals and humans are driven by feelings. There are no genes in the primordial soup. And evolutionary theory ignores consciousness and feelings. Darwin wrote about it a lot, but didn't really include it in the theory. So the question is, did feelings drive evolution? And this is also being studied in philosophy. It is known as the phenomenal argument, phenomenal experience driving evolution. And I would agree with that. I think that's might have been what happened. And this is a corollary that came out of the ORch-OR theory, which Roger may not agree with, but actually I've been discussing with him recently, and maybe he does. But it's a possibility and would explain a lot that feelings have been driving evolution. So I'll conclude by saying, number one, that quantum vibrations in microtubules support the ORch-OR theory by this formula E sub G equals H over T. They are testable and therapeutic targets for mental and cognitive disorders. If these vibrations are necessary for feeling good, we might be able to manipulate them with things like transcranial ultrasound, which could resonate or vibrate microtubules. Anesthetics prevent consciousness by dampening quantum terahertz vibrations in microtubules inside brain neurons, and psychedelics may enhance such vibrations, make you more conscious in a sense. ORch-OR provides temporal non-locality, which can rescue conscious free will and provide temporal binding. In quantum effects, you can have temporal non-locality, and we seem to get information from the near future in everyday activities of the brain. Cortical layer 5 pyramidal neurons are the best site for consciousness, large pyramid-shaped arrays of mixed polarity networks that can account for EEG. And there are no pyramidal cells in the cerebellum. The cerebellum is famous for not being conscious, despite having more neurons than the cortex. And ORch-OR addresses the hard problem of why we have phenomenal experience through quantum pan-protopsychism by saying that feelings are primitive and arise from collapses that are occurring ubiquitously around them, but when organized give rise to our full conscious experience. So happiness would be optimizing feelings and optimizing resonance and to drive evolution. Thank you very much.
Thank you, Professor Hameroff, for taking time out and then giving us this fantastic presentation. And we would always look forward for you to visit us and spend some happiness or share some happiness of yours with us. Thank you very much.
Thank you.